Good afternoon, Dr. Dan Guerra here with Authentic Biochemistry video lecture. Today is the 16th day of March, 2022. We're going to continue with our second in a series on diabetes. And what I want to do is hopefully cover a fair amount of ground so we can get to maybe only one more video lecture and then probably a couple more audio. But we've been spending a lot of time on diabetes, but it's well worth it. So let's leave Pedo Lake in beautiful British Columbia, Canada, and let's move on to the lecture. All right. Now, I think we covered some of this just recently on the audio, but I want to do it again very quickly on the video. This comes from a paper you can see published in Current Medical Chemistry. It talks about the insulin receptor substrates 1 and 2. Now, these are going to be the substrates for the insulin receptor, and they function in the liver. They also function in skeletal muscle. I'm going to show you a diagram about that. But let's go through the text first. So there's two different isoforms, 1 and 2, and they're probably the most relevant representatives of those kinds of proteins, although there are other isoforms, but these are the ones that most often have been researched and we know more about them. And what they do is they play what says here critical, they're involved in critical nodes in insulin and in insulin growth factor 1 signaling. So, sometimes the peptide IGF-1 essentially replaces insulin in certain cellular and tissue and organ environments. Particularly IGF-1 is really important in the central nervous system, for example. But at any rate, the receptors for these proteins, insulin or IGF-1, function through downstream multiple networks. And one of the major networks of this is this IRS sequence, right? The insulin receptor substrate sequence. So they're quite similar, but their structural and functional features would show tissue-specific functionality. So IRS1 is crucial for normal growth and differentiation of myofibers, obviously skeletal muscle, and it's insulin-dependent glucose uptake, and it also involves glycogen synthesis. So insulin is responsible for glycogen synthesis as well. If you remember glycogen synthase, right, an enzyme and the regulation of it. Now, so the presence of IRS2 in skeletal muscle is actually pretty negligible, and if indeed it has a function, it hasn't been worked out. But it's not involved as it appears because there have been knockouts. Now, this is in the mouse model for insulin-induced glucose uptake, and seems like IRS2 is not involved, at least in that model. And we talked about the caveats about these models because of the fact that we're dealing with different kinds of mammals and different kinds of digestion of carbohydrates, for example. That plays a very significant role in insulin signaling, as it turns out. So in liver, we have both forms, and they're important to mediate insulin-dependent regulation of glucose and lipid metabolism. And that's no surprise to you that have been listening to Authentic Biochemistry, because you know that the foundation of bioenergetic signaling and membrane trafficking is going to be in lipid metabolism. But basically, that covers most of the ground of what cells are required to do, right? So when you have a malfunction in metabolism, such as in diabetes, it's not much of a stretch of the imagination to understand that you're going to have an immediate malfunction in lipid metabolism. So diabetes is actually a dyslipidemia. And the fact that you can measure circulating glucose is why it's called diabetes and why it's normally recognized as a glucose disorder because of the whole aspect of insulin uptake. But really everything downstream from that, and even probably prodromal to insulin insensitivity, shall we say, in type 2 diabetes, which is what we're spending most of our time on, is really related to dyslipidemia associated with a lack of nutrient balance. And often, yes, that involves this horrible pandemic known as obesity. Okay. So dysregulated muscular or hepatic abundance and or the phosphorylation status of either of those two proteins become important factors in the pathogenesis of IR, type 2 diabetes, and also indeed in muscle wasting. Okay. So got that cleared now. So here's a diagram, which I want to spend a few minutes on. Again, we covered this sort of in the audio lecture last night, but I want to make sure you see it. So the muscle, insulin binds to its receptor, and it triggers the IRSs 1 and 2. And I told you the 2 is kind of not sure what it really does, but let's just say the IRS signaling system. And that will induce the phosphatidylcholine 3 kinase, which will phosphorylate protein kinase B. Protein kinase B then will trigger the mobilization of the GLUT4, that's the glucose transporter, from endosomal compartments to the plasma membrane in the muscle. Hence, that's insulin-dependent glucose uptake, you understand. Now, notice in muscle, if there's free fatty acid, that free fatty acid, as it's being taken up by the muscle cell, will induce protein kinase C theta. Okay? And that will act because of phosphorylation of a certain site on the IRS1-2, differential phosphorylation, of course, that will block that pathway. So blocking that pathway will block the mobilization of GLUT4 to the surface for glucose uptake. Plus, free fatty acids will immediately corrupt the glucose transporter itself. Where do you get a lot of high circulating free fatty acids or associated with serum albumin? That's correct, obesity. Right? Obesity is dyslipidemia, and it also means high levels of circulating fatty acid, and this is the major cause of what ultimately results in insulin resistance and full-blown type 2 diabetes. Now look at the liver. The liver, you have GLUT2, not GLUT4, so that's not something that needs to be mobilized by insulin binding. So in the liver, you don't really have insulin-dependent glucose uptake. However, you do need insulin to trigger IRS1-2, to trigger phosphatidylcholine 3 kinase, to phosphorylate protein kinase B. And what that protein does then is allow for transcription factors to function to increase glycolysis by increasing the expression of glycolytic enzymes, such as phosphofructokinase 1, phosphofructokinase 2, glyceraldehyde 3-phosphate dehydrogenase, aldolase, enzymes like that. And at the same time, tank gluconeogenesis. Okay? So that is what the liver is supposed to do with high circulating glucose, which is where the insulin secretion would have occurred from the beta cell of the pancreas. So you got insulin being secreted because you have high circulating glucose, like say postprandial. And what you want the liver to do is not synthesize glucose, because there's no need for it. What you want to do is metabolize glucose via glycolysis to make pyruvate. Pyruvate then can be converted to oxaloacetic acid and acetyl-CoA and run the TCA cycle. And with the high enough levels of NADH and FADH, most of that carbon, or probably a good 80% of it, will be converted after citrate with the ATP citrate lyase reaction to fatty acid. And this back in the cytosol, the acetyl-CoA will reconvene, and the OAA will be also produced there. And so you'll be able to make sufficient amounts of fatty acid for triacylglycerol synthesis, phospholipid synthesis. Triacylglycerol phospholipids, as well as cholesterologenesis, will all be loaded onto very low density lipoproteins in the liver and be trafficked out, and a lot of that lipid will be put into the adipose, right? That's how you convert carbohydrate to lipid and move it from the liver to the visceral fat or to any depot fat compartment. Okay. You can see here, same thing with high circulating free fatty acid, as in an obesogenic state, you're going to get a blockage of that glucose uptake. So what's going to happen when you block glucose uptake? You're going to get hyperglucosemia, which is exactly why it's called diabetes, right? So you get it from the muscle and from the liver. Adipose has a similar effect because the adipose also has insulin-dependent glucose uptake. A lot of free fatty acid in the adipose when you have a lot of adipose, right? Okay. So I think I made my point. All right, so let's go to the next slide. So here's dyslipidemia, a good definition about it. High circulating fatty acids are common in obesity. Obesity is a major epidemic, pandemic, really worldwide. Fatty acyl-CoAs, remember those thioesters, block glycolysis in the liver by increasing ATP production because of beta oxidation. That means you're going to get NAD and FAD reduction, and those are both reduced during the oxidation of the fatty acyl-CoAs. You're also going to be making acetoacetate and beta-hydroxybutyrate, the ketone bodies. High NADH also blocks, of course, because you know intracellular control over metabolism, the pyruvate dehydrogenase, glyceraldehyde dehydrogenase, and that will further inhibit glycolysis, right? Those are two enzymes in the glycolytic pathway. All right, so low glucose utilization increases serum glucose. Let me move my wonderful video image there. So low glucose utilization increases serum glucose, which increases pancreatic insulin secretion. So you get more insulin. So finally, that leads to insulin insufficiency and resistance. Because remember, you start getting damage to those islet cells, those beta cells in the pancreas, because of excessive amount of signaling through them, an excessive amount of insulin production and secretion. So ultimately, you get insulin insufficiency and ultimately insulin resistance at those receptors for the same reason, high levels of free fatty acid. So you see how this comes together, how dyslipidemia is the real pathobiochemistry of type 2 diabetes. So remember, when you're overweight or you have a high BMI, you're going to produce some pro-inflammatory cytokines, for example, TNF-alpha. TNF-alpha and free fatty acids are going to be produced because of a lot of lipase activity. That will both of those will lead to insulin resistance. Insulin resistance can then be recognized as presentation for the metabolic syndrome. Pancreas tries to compensate for that by increasing the amount of insulin. So you get insulin buildup, insulin elevated, blood sugar stays normal for a while because of the increase in insulin, the increase of insulin uptake in the skeletal muscle and in the adipose, where most of it is occurring. You still have high levels of circulating triacylglycerol. You get low levels of high density lipoprotein because you're not mobilizing the apolipoproteins out of the very low, intermediate, and low density lipoprotein fractions, which are also in circulation. That's because you're getting a buildup of triacylglycerol accumulation in the blood and the lack of deposition to the adipose. It's slowing things down. That will, of course, increase blood pressure because you're starting to get a kidney involvement. And the kidney cannot also take high levels of circulating lipoproteins or high circulating triacylglycerol or high circulating free fatty acid. Same kind of disorder that happens in the liver happens in the kidney, you understand. Eventually, glucose-free fatty acid and insulin will build up. Pancreas can't increase the amount of insulin anymore. And plus, insulin resistance comes on because the receptors aren't functioning because of the free fatty acid. And then you get type 2 diabetes. Again, insulin's higher than normal. Blood sugar remains high. Triglycerol is high. High density lipoprotein remains low. So again, you're not moving either cholesterol or cholesterol esters, so you have the potential for oxycholesterol or oxysterol synthesis because of the residence time of cholesterol in circulation and docking into various subcellular sites, including being taken up directly by macrophages and then forming foam cells. And of course, your blood pressure remains high. So it's all very bad, very high morbidity disease, type 2 diabetes, you see. So remember what the regulation of glucose metabolism entails. Insulin is synthesized in the pancreas by the beta cells. Those are called the islets of Langerhans, right? Beta cells produce proinsulin. Alpha cells produce glucagon. That's during fasting. Delta cells produce somatostatin, which sort of is a hormone that controls both insulin and glucagon transcription. And then you have something called F cells, and those produce the pancreatic polypeptide. that has more to do with digestion. Okay. What about the hormonal regulation? Ingestion of nutrients stimulates the release of glucose-dependent insulinotropic polypeptide, that's GIP, and the somewhat more difficult to remember, the glucagon-like peptide 1, or the GLP-1. So you have GIP and GLP. And what happens there is from the cells in the gut, these proteins tend to stimulate the production of insulin and inhibit glucagon, which is what in other words, they're aiding in insulin-dependent glucose uptake and insulin clearance of circulating glucose. So insulin stimulates the diffusion of glucose into adipose and muscle tissue, as we've already covered. And glucose oxidized in the cell, that is the glycolytic pathway, also should be occurring. And then that's used primarily in the liver. You can use that glucose for glycogenesis, but not necessarily for glucose, okay? So not for gluconeogenesis, but for the synthesis of other carbohydrates as those that are used in glycoprotein biosynthesis. And what are the glycoproteins? Many of the receptors that are in the cell, as well as some of the apolipoproteins, cytokines, chemokines, metalloproteinases, and all the other proteins that get secreted. Immunoglobulins are another glycoprotein, of course. Okay. So, the fasting state, glucose is produced by glycogenolysis, right? Because glycogen phosphorylase is triggered by this response, right? If you recall. And gluconeogenesis, well, insulin secretion falls to basal level, Okay. Glucagon then is responsible for the most glucose production in the fasting state because basically it involves glycogen breakdown in the liver and then helps glycolytic pathway get that gluconeogenesis along with glucose 6-phosphatase to release glucose from the liver, right? This is what happens during fasting. This is how you get glucose back into circulation. Now, there are other hormones involved, corticosteroids, growth hormone, catecholamines. And all of those hormones tend to maintain, or as they say, augment gluconeogenesis or glucose release via glycogenolysis. Okay? This is in the fasting state. Exercise also plays a very significant role. Initially, insulin levels drop, and glucagon and catecholamine levels increase, increasing the production of free fatty acids because of hormone-sensitive lipase activity. And that will stimulate glycogenolysis, breakdown of glycogen because of glycogen phosphorylase, that enzyme which is controlled by glucagon and catecholamines. Okay. So the rise in glucose is used then to meet the initial primary energy needs. And here you're primarily talking about the central nervous system. The cardiovascular system also requires glucose, but it also spends most of its bioenergetics on fatty acid. Anywhere between 50 and 80% of normal heartbeat is done with fatty acid oxidation, not with glucose. Yeah. But central nervous system, it's much better than 90% glucose utilization. Right? After that, you get ketone bodies and some amino acid transamination reactions, making keto acids and running the TCA cycle, doing that kind of NADH, FADH production, and ultimately electron transport for ATP synthesis in the intermitochondrial membrane. So muscle tissue increases the metabolism of glucose, obviously, because this is going to be where a lot of other glycogen is stored, the liver and in skeletal muscle. And you're going to need all that glucose to be able to do muscle contraction, right? That requires glucose oxidation. And so as exercise continues, increasing insulin sensitivity is good because insulin then, as soon as it is secreted, will be immediately sensitized at the right molar concentration to bring glucose out of the circulation and back into the muscle. Okay. So that maintains, and of course you're going to get gluconeogenesis in the liver via Cori cycle and alanine cycle. Now that glucose is going to be taken right back up into muscle tissue. Right? Because the muscle is the one that's going to be competing with the adipose tissue. And because there's a lot of contraction going on in the muscle, you're going to be able to get adequate amounts of glucose to that muscle so it continues to function. Right? And that occurs in the presence of low insulin levels because that's where you're going to be because you're going to have low circulating glucose. Now, stress is another component. So stress, you get a production of the stress hormones. We know these as corticosteroids and the catecholamines, which are adrenaline and noradrenaline, right? And these increase the production of glucose in the liver, again, and glucagon in the pancreas, and they decrease the utilization of glucose. So stress hormones is going to decrease the utilization of glucose so they can be readily available for the fight or flee response during stress. You understand? That's what this is going on. That's what's going on here. So the catecholamines increase the production of free fatty acid release, and they inhibit glucose uptake in the periphery. So the central nervous system can continue to function on free circulating glucose during stress. All those events ultimately lead to hyperglycemia during stress because you're getting a lot of glucose kept in circulation, right? Because you're not getting that much glucose being taken up because insulin levels are decreasing during stress. So the classification of glucose intolerance disorders are type 1, type 2 diabetes, mellitus. And then there are other types of diabetes we covered in lecture already, diabetes type 3, remember that one. And then there's gestational diabetes that occurs during pregnancy. Yeah. We've talked about all these previously, but here I'm just listing them. So type 1 diabetes is characterized by destruction of the beta cells of the pancreas, an autoimmune disease. It's diagnosed when children are relatively young. Sometimes it remains occult all the way to people in their late teens or early 20s. Usually, though, most people that have enough destruction of the beta cells are going to be insulin dependent. And so this is going to be picked up in pretty young children, way before the age of 5. But in rare cases, this cannot fully develop and present until people are a little bit older in their teen years. The etiology, of course, is immune-mediated. Some say it could be idiopathic because sometimes you don't see autoimmune markers like a lot of pro-inflammatory cytokines. And there is some association with the human form of the MHC class 2. That's the HLA, right? That's the protein that's on the surface of antigen-presenting cells. And that has something to do with this autoimmune response. The different subclasses of HLA allowing for the beta cells to look like cells that need to be attacked by both leukocytes and lymphocytes, therefore inducing the autoimmune response, killing the beta cells, and decreasing the insulin secretion altogether. So the result is finally, of course, you get insulin insufficiency, so you have to take insulin in. The overproduction of glucagon without insulin counteracting this will stimulate more glycogen breakdown and more glucose synthesis from non-glucose intermediates. So you're going to get glycogenolysis and gluconeogenesis. Glucose levels will rise according to this paradigm, and that can lead to polyuria because of the kidney malfunctioning of all the high circulating glucose, polydipsia, and polyphagia. These are all part of the initial disorders associated with T1, type T1D, type 1 diabetes. Free fatty acids, because there's a lot being produced to keep up with the requirement for bioenergetics, are going to be generated because of hormone-sensitive lipase, and those are going to be transformed into ketones after beta oxidation in the liver and the ketogenesis. So a lot of acetoacetate and beta-hydroxybutyrate in circulation. And sometimes these ketone bodies can lower the pH eventually, because you're also going to get a lot of lactic acid in the blood. And all of that can lead to a lowering of the blood pH, which can cause a severe disability in type 1 diabetes. This is why lack of insulin can cause insulin shock, and that's entirely because of the lowering of the blood pH. Yeah. So see here in the GI tract, you've got glucose coming in, you've got ketone bodies going into neural tissues, you've got glucose going to neural tissues, right? GI tract is releasing glucose. This is presumably from nutrition. What is the liver doing? the liver is taking up free fatty acid. It's producing ketone bodies, right? It's also trying to produce glucose via gluconeogenesis from, say, lactic acid or from alanine after transamination to pyruvate, of course. And you're also getting glycogen breakdown, right? Pancreas is trying to produce glucagon because of the lack of any circulating glucose. Insulin secretion is going to go down, right? Because glucose levels are going to also be going down, even though the liver is trying to pump it out for the CNS, right? Muscle tissue is going to be picking up free fatty acid because that can be picked up by CD36, for example, one of the receptors, orphan receptors, and that allows for that fatty acid then within the muscle to be used as acyl CoAs for beta oxidation as long as there's enough molecular oxygen. But also you make oil droplets in the muscle even during the well-fed state, and those oil droplets will be converted to, after triglycerol lipase activity, to fatty acyl CoAs, run through the beta oxidation pathway, and make ATP that way for the contraction of the muscle. Okay, but free fatty acid will get taken up by the muscle tissue. Adipose tissue is going to be producing a great deal of free fatty acid because of lipase activity, basically. So this is how you compensate for low circulating glucose. These are called glucose intolerance disorders, right? And that's what type 1 and type 2 diabetes actually essentially are. So type 2 diabetes, then. It is now the most common type of diabetes. It wasn't until really the early 1960s. It started to become more dominant. Now it is the dominant. Now it's a major comorbid disease associated with obesity, something we've been talking about now for several days. Non-Caucasian people and elderly seem to be disproportionately affected. So there's some racial and also some age and even some possible sexual dissimilarity for type 2 diabetes, with more men getting it than women. But it could be because more men tend to put on more visceral fat, and visceral fat accumulation leads directly, or not directly, but can lead very efficiently to type 2 diabetes. Common risk factors, therefore, for type 2 diabetes are obesity, aging, and sedentary lifestyle. If you have a sedentary lifestyle, you're not doing a lot of muscle contractions, so you're not maintaining the normal control over glucose homeostasis by burning that glucose, storing glycogen, or storing that carbon as fat, which can then be mobilized during the long periods of fasting. Sedentary lifestyle normally is equated with a lot of non-fasting, regular high caloric density intake. And this is what also leads to obesity. Insulin resistance, of course, then kicks in. Beta cell dysfunction starts to occur for the reasons I've already explained. And that leads to a relative lack of either insulin itself. This is the later stages of type 2D, going from prodromal to full dromal state, but also because of that problem with fatty acid, corrupting insulin sensitivity, right? Causing insulin resistance. So here, the GI tract will produce glucose. The liver will continue via glycogenolysis and gluconeogenesis to produce glucose. The neural tissue will be taking in glucose still because you haven't really produced a lot of ketone bodies. Pancreas will produce insulin, but very low levels. It'll produce a lot of glucagon because, again, the circulation is going to be low relatively in glucose. So glucagon is going to be used for, again, the liver for generating glucose synthesis or glycogen breakdown, both of which would give you net glucose into the serum. Muscle tissue continues to take up glucose, and so does the adipose, because you have adequate amounts of insulin to be able to do this. Right? Plus, you have multiple transporters in those two tissues, so you'll always be able to pick up some glucose, even when insulin levels are not particularly high. So with type 2 diabetes, you also get polyuria, polydipsia, and polyphagia, but they're probably more subtle than what you get in type 1. You get ketoacidosis, but it's very uncommon because there isn't a lot of fatty acid breakdown or ketogenesis in type 2 diabetes. You get hyperglycemic, hyperosmolar, non-ketotic coma, and that can develop due to severe dehydration, and it's more commonly found in older adults. Right? It's very dangerous because if you get HHNK, it's not diagnosed, you can go into shock and you can actually have, before coma sets in, it's also potential to cause stroke, because of embolism being generated because of lipoprotein accumulation in the arterioles. And when that happens, stroke and also can lead to kidney damage and, of course, the central nervous system damage. So it's a very dangerous disease. The other types of diabetes are some kind of defect in the beta cell. So you have mature onset diabetes of the young. That's called MODY. It's kind of rare these days when it's diagnosed. You can get genetic defects for insulin action. That means something's wrong with the insulin receptor or the insulin receptor substrate, or maybe with the protein kinases like the phosphoinositol 1,3 kinase or the PKB, right? So this would be downstream signaling of insulin. Sometimes you get those as genetic disorders. Relatively rare. You can also get a whole host of endocrinopathies. This has to do with leptin, adiponectin, as well as the catecholamines, and then all of the corticosteroid hormone synthesis coming from the POMC and whatnot through the HPA axis, right? And drug, chemical infection-induced diabetes. This is from illicit drugs, usually something like cocaine, heroin can give you a certain kind of diabetes. This really has to do with a lack of function of the entire system, bioenergetics, hormonal signaling, as well as membrane corruption. Alcohol also is a neurotoxic and cause similar problems, or multiple infections can also sometimes induce sort of a type 2 diabetes, not a type 1. But even type 1, if you get enough pro-inflammatory cytokines starting to induce an autoimmune response. Gestational diabetes can occur in somewhere around a quarter of all normal pregnancies. But it comes and goes. It doesn't usually last throughout the entire gestation cycle, but it can be dangerous for women and for the fetus, the baby. It's a disorder of glucose intolerance, variable severity, as I said, and the onset is usually during pregnancy. Closely resembles type 2 and all those different phenotypes I just told you about. Most likely precipitated by the presence of placental hormones. Again, it's an endocrinopathy is what's going on. And the management is through dietary counseling. This is basically control over carbohydrate uptake in the diet. More exercise, too, during gestation, and keeping that blood glucose to ketone level ratio at a good stoichiometry. Okay. So with the diagnosis of glucose-dependent GDM, that is just standard glucose disorders, usually of the diabetic type, right? Fasting blood glucose levels that are about 95 for 100 grams of glucose load. After 1 hour, it goes up to about 180. Then it drops down to 155 and 140 in 3 hours. If you have a lower glucose load, you can see it starts at around 95, and then it also starts to decrease. So 2 or more. So if you get your blood drawn after a glucose tolerance test, which is what you would do if you were going to the, if you were going to find out whether or not you had diabetes, okay? You wanted to know whether or not that was the case. 2 or more blood glucose determinations be equal to or greater than those values shown there to establish a diagnosis of some kind of glucose intolerance. That's what that means. A glucose intolerance test is the first way to determine whether or not you might have diabetes, right? Test has to be done in the morning after an overnight fasting and between 18 and 14 hours, and after at least 3 days of unrestricted diet, which means at least 150 grams of carbohydrate per day. So you can see this is what's normally done in the clinic in an outpatient setting. Unlimited physical activity is allowed, of course, because it's not going to be involved in the short-term glucose intolerance tests. And again, this comes from right out of the American Diabetes Association, which is part of the NIH, actually. Pre-diabetics, you can usually be screened for that. You get impaired glucose intolerance, that is, you get glucose intolerance, and you get impaired fasting glucose tolerance. Both of those are glucose intolerance. Intermediate stages between normal glucose metabolism and diabetes can also be picked up during these phases of these exams. And what it does at this stage, at the prodromal state of T2D, or usually T2D, because usually T21 is already discovered, or for gestational. It represents a risk factor for the development of diabetes, is basically what it is. And if it lasts a long time, even as a prodromal state, it can start to cause cardiovascular problems. And this has to do with cardiac hypertrophy, fibrosis, also some liver damage, inducing some dyslipidemia, which can also alter heart function, and of course, atherosclerosis, which has to do again with those high levels of circulating lipoproteins, some of them incorporating along with cholesterol esters into macrophages being picked up by diabetes into the arterial walls, say, of the cardiac muscle, and this can lead to then atherosclerosis. Right? And so you have to be very careful with even prodromal phases of diabetes, because it starts a long pathway into a history of morbidities, which can ultimately get you to be wheelchair bound and really sickly in your 40s or 50s if it happens early. So obesity really needs to be checked very early in life. Is to be basically dealt with before you can hope to deal with any kind of underlying carbohydrate lipid problem, because that's what's causing it. Right? Some manifestations and complications, of course, you can get acute hyperglycemia because of the lack of glucose uptake, right? Because of insulin sensitivity. It's caused by alterations in nutrition, inactivity, inadequate use of anti-diabetic medications if you happen to be on those, like metformin or thioureas. Symptoms include, again, polyuria, polydipsia, polyphagia, nausea, fatigue, blurred vision. And you also are more prone to infections because of all the corticosteroid circulation, which actually works to inhibit the immune response. Right? And that's not what you want. I think I'm going to stop there. Because I don't really want to get to the whole discussion of further manifestation of type 2 diabetes until we get back to some biochemical considerations, some pathobiochemistry, because this right now is going to give us a lot of pathophysiology, and I can do this straight up on the audio lectures, right? So I'm going to stop here. And again, I'm going to ask you to, anytime you have any questions for me on the video, YouTube, go ahead and post them there. I try to answer most of them. Some of them, if they're difficult for me to interpret, I might just let pass. But I'm also looking for people to come to these YouTube lectures and to give me some idea of how many people are actually listening to them. And what I would like to do is expand out the video lectures in authentic biochemistry to do standard biochemistry, general biochemistry lectures for a larger audience. So I'd like to do that at the same time, contemporaneously, with doing authentic biochemistry, where we do the primary research literature, right? Which is what I mostly do on the podcast, but I also, of course, do in the YouTube video lectures. Either way around it, I want to hit the primary referee literature. That's my main function here in authentic biochemistry. But I'm glad to do these general biochem lectures as well, so we can all keep fully aware of the multiple metabolic pathways, changes in gene expression associated with disease state and aging, and also immune responses. So I'm going to stop there. Thank you very much for paying attention. Again, this is the 16th of March in 2022. So we're getting very close to spring, and the weather's starting to look really nice out here in the Pacific Northwest. And I hope it is okay for where you are. So this is Dr. Dan Guerra from Authentic Biochemistry Studios saying goodbye for now.